talking about how protein signal calcium is the second messenger, and we finished that last time. Excuse me. Sorry. Talked about that calcium by itself doesn't do anything. It binds to other proteins and causes them to do things like that. So that signal is really important. If PKC, for example, gets turned on the wrong cell type, it causes rapid cell division and can form tumors. There are several different mutated forms of protein kinase C that cause different kinds of cancer. So it's really important to control the signal. And if you look up here, these are all ways or mechanisms to reduce the calcium signal once it forms. So this is the exchanger that we talked about, the antiport. This is a calcium pump. But how do you get calcium into the cytoplasm? We talked about one mechanism, and that was the IP3 receptor. If you look at the outline, you realize there's another protein just like the IP3 receptor that does the same thing in skeletal muscle. Now, why is calcium important in skeletal muscle? It causes contraction. It's called stimulus contraction coupling. Okay, so, but it doesn't use the IP3 receptor. It uses a very similar protein that's not gated by IP3. It's gated by a chemical called ryanodine. Okay, so make sure you look at that. In skeletal muscles, ryanodine receptors are what open up, and it's a CICR, it's a calcium-based calcium release channel, just like the IP3 is, but it's not gated the same way. In fact, in some ways, it's actually gated by a membrane protein that sits up here that's called the store-operated channel, okay, store-operated channel. And what it does is when it detects a change in membrane voltage, or it's gated by various different ways, but when it opens, calcium can come in from the cytoplasm very rapidly. So store-operated channels, another way to get in. That's in the outline, it's not on this picture, so that's why I'm pointing it out. Another way is just a classic voltage-gated calcium channel. That's not a store-operated channel, it's gated only by voltage changes. Okay? And we looked at an example of that last time. That's actually how a cardiac myocyte gets its strength of contraction increased, because it gets phosphorylated by pKa, and that channel is a voltage-gated calcium channel. So those are all mechanisms that are up here that allow the signal to come in. And then these guys, plus ones that we talked about last time, like these calcium binding proteins, calbindin, calsequestrin. This is a calcium ATP. This is circa. So these are all off signals. Okay. So put the two categories in two separate places and know which ones control what. Okay. All right. So we talked a little bit about calmodulin, and so this thing binds to calcium. It detects the calcium signal, wraps itself around like a kinase, and then activates it. So this is actually, this happened, the form of this enzyme, calmodulin kinase 2, that lives in the brain, is actually very important in memory, okay? And that comes in handy sometimes. So this is the inactive form of CAM kinase 2 in neuron. And so it actually, a lot of these kinases actually go around with a foot in their mouth. Okay, so this is an inhibitory domain up here, and it's blocking the catalytic domain that puts a phosphate on serine and threonine. This is a serine threonine kinase. Okay, how do you get this to turn on? Well, what happens is there's a pulse in calcium. There's a really nice video in your books, a DVD it just shows a nerve cell, and it's just flickering because these little calcium pulses are going through the axons. And so that calcium goes up, that actually binds to calmodulin, and now it's ready to bind to its target. And its target is this thing. And so it wraps itself around, so the N-terminus and C-terminus become very close together. And we can actually follow how close together they are by putting fluorescent probes on there, okay? And what happens is it nudges this enough, dislodges, look what it's doing. It's blocking the inhibitory domain. So this thing gets a little bit of activity, and what's the first thing it does? It phosphorylates itself in neighboring CAM kinases. So it's going to phosphorylate itself, and it's going to phosphorylate neighbors, And so you get this spread of this memory molecule, okay? And the memory has two bases. The reason this will stay active, and what it's doing, is remembering the calcium pulse just went down that axon. And so this is molecular memory. And this stays on and remembers that the calcium is there, because as long as calmodulin stays bound to the inhibitory domain, the kinase can go around and phosphorylate proteins. And it can cause synapses to grow, which is another basis of memory. So this is a really important protein in driving building of memory traces, okay? And even after the so what makes this release? Calcium signal will fall, okay? And eventually, if that happens, the calcium will dissociate from calmodulin. This thing will unwrap. But what's cool is the memory still lasts a little bit longer, because calmodulin has converted this into an open form, so that it still retains, as they say here, 50 to 80% of its kinase activity. So it's still remembering the calcium trace. And so they think this is very important. And many times a memory, what happens is if you take a mouse and you knock out this gene for this CAM kinase 2, they have trouble remembering where the neighborhood CVS is. And so it's really directly related to, are you awake? It's a joke. Okay, so they have little memory things that they go through, and they forget how to do it. Okay, so we know it's involved. All right, so the other thing is these pathways, signaling pathways, crosstalk. They talk to each other. Okay, this is a perfect example of how the cyclic AMP pathway is going to be modulated by the calcium pathway. Okay, so let's say that you have G sub S, let's say epinephrine binds, you know the whole story, you end up getting a pulse of cyclic AMP. You know that to turn it off, it's cyclic AMP phosphodiesterase. Well, what you might not have known is that they're calmodulin dependent phosphodiesterases that turn this off. So it's the calcium pathway turning off the cyclic AMP pathway. Can they turn each other on? Absolutely, they can. Okay. So it gets very complicated when you get crosstalk between two pathways. And what about 5? What about 10? So it forms a network that's very difficult to decipher, but it can be done with different modeling techniques. Okay, here's another, since we're talking about some off switches, that was one off switch. Here's a different off switch, and that is, this is all called receptor desensitization. Everybody knows what that means. If you're desensitized, you don't feel anything. In the insulin signaling world, we call it insulin resistance. It's resistant to the insulin signal. And so all of these mechanisms are applicable to lots of different diseases that involve some sort of pulling off or inactivation of the receptor. So let's look at the various ways that can happen. We're already familiar with a few of these, okay? So if you sequester the receptor, okay, what that means is the protein receptor is pulled off the cell surface. You know how that works? By receptor-mediated endocytosis, right? If it goes to the endosome, it's not going to be destroyed as long as that endosome is what? What's the first endosome it should go to if it's not going to be destroyed? Early endosome, okay? And then what's missing? If it's going to be recycled, I just gave you the answer, it has to go to the recycling endosome, and then it goes back to the cell surface. But it does hide this for a while, so you can kind of make the cell quiet down. And this kind of thing happens in neurons, where they have to be excited and come off, be excited and come off. Okay, so this receptor hiding, or sequestration, is an active way of desensitizing the cell. Now, if you want to be more blunt about it, and this usually happens to like growth factor receptors, those opiate receptors, the dangerous receptors, what you do is you send it to the early endosome, and then it goes to the late endosome, and then it goes to the lysosome, and it's destroyed. It's going to take a while for the sensitivity to recover, because you have to turn on the gene and make more protein, right? Okay, so that's a slow, it's really going to turn it off for a while. Now, can you inactivate the receptor? Have we talked about ways that you can inactivate the receptor in any signaling pathway so far? What's a good way to, well, we've talked only about G protein coupled receptors. So how do you turn a G protein coupled receptor off? Not the G protein, the receptor. There's a whole class of proteins that do that. They're in fraternities and sororities. The Greeks, okay? They're G protein coupled receptor kinases. GPC receptor, or better known as Greeks, okay? And so if they phosphorylate the third intracellular loop or the C terminus of the G protein coupled receptor, they block the ability of that G protein to be activated by it. So phosphorylating this is a great way to turn it off for a little bit, desensitize it, okay? Now, what's going on here? This is the intracellular, the signaling protein, the transducer, so that would be represented by adenylate cyclase. It can be represented by the G protein. How do you turn the G protein off? First of all, can it turn itself off? No, it needs a gap. That's the inhibitory protein. The gap is going to activate. The activity is there. You're correct about what you're thinking, oh, well, it's a small GTPase. Yeah, but it needs to be kicked in the pants in order to activate its GTPase. And so the thing that does that is a gap, right? But in this case, there's a specific class of proteins. We don't call them gaps. We call them RGSs, regulators of G protein signaling. And on the test, you're going to get GPCR and Greeks and RGS. You're not going to get the word spelled out for you. So make sure when you make a card for those people, those proteins, that you know the abbreviation, not the spelled out form. If it helps you learn it, obviously that's good. But I'm not going to put regulators of G protein signaling. Is that something that regulates G proteins? It answers itself. Okay. RGS proteins are going to do that. Right? What about adenylate cyclase? That's a transducer. It's a transducer because it's not regulating, it's not activating a protein, it is producing a second messenger. Okay? Just like an IP3 receptor is a transducer. It produces the second messenger. Can you turn off adenylate cyclase?
You just said there was the what was it? The calcium calmodulin regulated adenylate cyclase.
So that indirectly, so that's a way of destroying the signal that adenylate cyclase. So that's indirect. But is there a direct way of doing something to the adenylate cyclase that will turn it off? There's actually two or three. Every time, well, let's take the most obvious case, the G inhibitory alpha subunit, right? Its job, when it's activated, is to go and activate adenylate cyclase. So that's the most obvious way that a protein will go turn off that right there, which is a transducer. Okay. And remember, the other way to get this to go off, if that's a G protein, is the more times it bumps into a target, it can hit about 100 different targets before it loses its activity. But every time it bumps into adenylate cyclase, if that's G sub S up here, it's going to be more likely to split its own GTP. Now, I think I confused some people with the G. This is not a RAS protein. This is not a small G. This is the alpha subunit of the heterotrimeric. So yes, it can split its own GTP. Sorry, I got it mixed up. But so this thing is able, without a gap, slowly to turn itself off. It becomes more likely to split its own GTP when it bumps into the target. Okay. Now, to clarify completely, if it's a small GTPase like RAS or RAB, it needs a gap. Okay. All right. Now, what about this third situation? So somehow the signaling pathway turns on an inhibitory protein. Okay. And that inhibitory protein, it could be Greek, okay, it could be a protein that we haven't talked about. It could be this protein, which is called arrestin, okay. And how this works is fairly complicated. So this is let's say this is the beta-2 adrenergic receptor. You know that that's the GEF for a G sub S, G protein, okay? Now, if you want to turn it off, you need to phosphorylate it in that third intracellular loop, or that little thing sticking out, that's the C terminus, so that must be the N terminus, okay? And so if the Greek will come up here and phosphorylate that. And then, yes, that inhibits the ability of this to talk to a G protein. But if you really want to block it, what happens is this protein is expressed and it binds to the surface, this intracellular surface of the G protein coupled receptor. And that arrestin tells you what it does. It arrests the ability of that receptor to activate G protein. So that's a great way of turning it off. It gets even more specific. I don't know if it's shown. No, I guess not. Arrestin, if it stays there long enough, will recruit AP2, and clathrin. Now what's going to happen? You're going to get one of these two possibilities. So it's either going to be pulled off the cell surface and hidden, sequestered, or it's going to be pulled off the cell surface and sent to the lysosome. So it gets very complicated how all these different mechanisms fit together, but this is a good example of what we call homologous desensitization. And what does that mean? It means the receptor that started the signal actually causes the production of a kinase that comes and turns the receptor off. It's all in the same pathway, therefore it's homologous desensitization. And there's this is actually, the Greek that turns this off is the beta receptor kinase, BARC, beta adrenergic receptor kinase. And so there's a very specific Greek, yes.
Is arrestin always associated with the Greek in order to turn this off?
It's not associated with the Greek, it's associated with a particular pattern of phosphorylation.
Well, I mean, is arrestin always needed to attach to those phosphate groups after Greek does its work?
This will turn it off for a short period of time. The problem is this, there's lots of, as you'll see in all these signaling pathways, we never talk about it, but there are all these constitutive phosphatases trying to dephosphorylate the phosphates you put on there. So unless you're actively phosphorylating it, it's going to be reactivated. And so in that case, this can be reactivated. But so if you really want to assure it's going to work, then beta, it's actually called beta arrestin, will recognize this phosphorylation pattern and bind to it and make sure it doesn't get turned back on. That's a good question. Okay, let's try a couple problems here. We're not going to do clickers because the machine's not working. Don't worry about it. Try to do this on your own first, and then we'll do it as a group. Okay, now get with your group and come up with the final answer. This should be fairly straightforward. Are you looking for a group? You got it?
Dr. Collins.
Yeah.
Yeah.
I'm sorry?
We're just looking for the also.
That's the gap. So remember those abbreviations, because you'll need them on the test. Because this came from the test.
Oh, I remember the.
Just remember it's a gap.
Yeah.
Okay, so how many groups have gotten their answer? Well, a couple more minutes. Okay. So let's just go through this. G protein coupled receptors activate G proteins. That's the GEF. You know? And upon binding to the receptor, the G beta gamma sodium then catalyzes the GDP exchange by the G alpha subunit. What's wrong with this statement? Okay. Is anything wrong with it? If you think there's nothing wrong, then you pick this one. And what part of this is wrong? Group 5. Lucky 5. Which group 5? Nobody's here today from group 5? Okay, you don't want to be. Okay, what part of that's wrong? So the G protein does bind to the receptor, it has to, because it's the GEF. That's the GEF. Does the G beta gamma subunit catalyze the GDP exchange? No, it doesn't. That's not what it does. It's actually inhibiting the whole process. Right? It's actually so tightly down to the alpha subunit, it can't let the GDP go. The role of the GEF is to open that thing up so that the GDP will leave, and this thing that actually needs to open up or dissociate completely, and then you can have the GDP exchange. And so the alpha subunit, the receptor stimulates the alpha subunit, not the beta gamma subunit, to exchange GDP for GDP. And so this is wrong because that has no GDP exchange activity. It doesn't bind GDP. This is wrong because the gap turns on the GTPase, which is an off signal, not an on signal. Okay. All right, so let's do one other, and this is also from former exam. Remember, trafficking is going to be a main thing on the test. I am going to get these practice tests to you early. We're almost 3 weeks out from the test. And actually, today's material will probably be the end of test quiz 3 material. And that's actually not that awesome. There's a lot of material here. But it's better than having cytoskeleton and cell cycle, which has happened some years. Okay. All right, so let's go to this one. Here's a simple question. Okay, so I'm not going to have you read this. I'll just tell you the upshot. Can I have your attention? Some bright person has invented a type of protein that will change the topology of a protein, okay, by binding to it and binding to these topogenic sequences. And so what happens is they're going to use this as a cancer treatment. And what we're going to try to do is, you remember what NDR1 looks like and does. Would that be a good thing to get rid of? Yeah, right? Little slutty transporter. Okay, so we want to do something that makes it inactivates, right? So what we're going to do is we put this drug in there that's going to flip its topology. So you need to know what the topology is. So it's useful to know the structure. Do you know where the N terminus and C terminus are in NDR1? Do you remember? It's got 12 alpha helices. N and C are cytoplasmic. We're going to flip that, okay? And so now the N and C are exoplasmic, and that just reverses the topology. Okay? If you want to draw it on a card, just go look at it. Okay? What is it about that makes it non-functional? Okay, not breaking the protein, but which thing is unlikely to be the reason it's not going to work? In other words, that's not a good reason to do this. So you're trying to find, what's going to make it not work, But which of these reasons is not the one that actually is going to cause it to become non-functional? There's one bogus answer. Most things in here are completely true. They will inactivate NDR1 because of something. I would draw it if I read it. And this is the way you ought to do a lot of these problems. If you don't draw it, you never know what you're getting correct. Let's see, NDR1. Draw it. What does the new topology look like? You can work it All right, so if you reverse the topology, the N-terminus and C-terminus are now exoplasmic, correct? And the parts that were exoplasmic last time are now cytoplasmic. So and someone was correct, you don't have to draw it, but it helps so that you don't miss simple stuff, okay? So, will that pump not be correctly glycosylated? It will not, right? It won't be glycosylated at all because those it has to be the exoplasmic domains that get glycosylated. There's no consensus sequence on the portion of the protein that can never be exoplasmic. So what's going to happen is all the consensus sequencing for glycosylation are going to be on the exoplasmic domain that's now cytoplasmic. So there's no hope there, okay? The pump's nucleotide binding domains will now be exoplasmic. Is that correct? Yeah, because remember, the rule is for a pump, those ATP binding domains have to be cytoplasmic. You've now made them exoplasmic. So that's a great way of screwing it up, okay? The pump will now have fewer transmembrane segments. I don't see any reason why you change the number, the same number of topogenic sequences are there. You just change the way it's inserted in. All you gotta do is change the way the first one is inserted and everybody follows. Okay? So that's the answer. But let's go through this one. The soluble binding site will now be incorrectly oriented. Yeah. It usually binds from the cytoplasm or from the lipid bilayer, and it faces and has a precise orientation, okay, in one of the different binding states. Okay, this one's a little bit more complicated, and you do have to know this. So this is a membrane protein. To get it to go from the ER to the Golgi, how do you get it to go there? You have to know the coding type, and so it is top coding, and the information for recruiting the coding is on what side of the membrane. This is not a soluble cargo, it's a membrane cargo. The sorting information has got to be cytoplasmic. You've now made that sorting information exoplasmic. How do you know that? Because the sorting information is on the C-terminus of a membrane protein as a cargo. That's very detailed. That's how detailed you have to know to set this trafficking stuff. Okay. And there's consensus sequence. If you're a soluble cargo, all you have to have is you have to have another C-terminal sequence. It's called the KDEL sequence. The KDEL receptor is the one that has to have this information. But if you're a membrane protein, your C-terminus has to have the machinery that will recruit the top two coding. Yes.
What if you have a protein whose C-terminus is exoplasmic normally? Would the coding information wouldn't still be there, right? It would be somewhere else.
Right.
Okay. So
Yeah,
you would
it doesn't belong in this.
in this case.
Right.
Okay.
Okay. Okay, so that's a complicated one, but it's as long as you know the way these things work, then you're going to be able to get it right. Just think through it. And I still recommend drawing it out, particularly if you're on a test and you're all fuzzed out. Sometimes drawing stuff makes it clearer. It makes it harder for you to make a small mistake. Okay. Let's take a brief break. Okay, let's go ahead and get started again. We're going to talk about cancer a little bit, but to do that, we have to answer this question. What makes the receptor tyrosine kinase family, and that's the new receptor family we're going to talk about, likely to be activated in cancer cells? So that's what we're after. To get there, though, we have to know what this family is like. So this is what they look like. And the reason they're called RTKs is because the receptor itself has a kinase activity. It's embedded in the sequence. That sequence must be cytoplasmic. So the N-terminus is designed to detect the ligand. So this is the epidermal growth factor receptor. And typically, all except for the insulin receptor family, is they're monomers. Okay? And it's the ligand that causes them to dimerize. But they must dimerize in order to do their stuff. And you notice the insulin receptor is already cheated because even without insulin, it's already dimerized. Okay? And so we have to ask, what's weird about that? So what happens is this will bind to epidermal growth factor. It will recruit another epidermal growth factor receptor, and they dimerize. Why is that necessary? It's because when this becomes active, it's going to cross-phosphorylate its buddy. It's going to take a ATP, pull off the gamma phosphate, and stick it on an adjacent receptor. So it's called autophosphorylation, but it's really cross-phosphorylation. Okay? And here's all the different ones that are involved in this pathway. But this is the basic outline. So you have inactive, the signal molecule binds, it dimerizes, and then there's cross phosphorylation. So they just load each other up with the phosphates. Now, there's two reasons for that. Number one is, like the CAM kinase, when it phosphorylates itself, it becomes hyperactive. So it actually phosphorylates the kinase domain and changes its conformation so that it becomes even more active than it was. And remember, it's very specific. It's putting phosphates only on tyrosine, only on tyrosine. The second reason is this is now a scaffolding protein that can recruit all the members of the signaling pathway to it. And it needs lots of surface area. Okay? So what kind of protein is it going to recruit? It's going to recognize a phosphotyrosine. That has to be the essential element. And those proteins have all one thing in common. They all have something right here in this domain. So these are all the signaling proteins that then go over and stick to the activated receptor. They all have what's called an SH2 domain. And SH2 refers to Sark homology 2. Okay? And clearly that refers to our old friend C-Sark. And it looks like a portion of the C-Sark molecule. And that portion is what recognizes the phosphotyrosine and then a few adjacent amino acids. So it's not looking just for any old phosphotyrosine. It's got two plugs, and that's often shown. Let's see if we can. So this is the binding domain for an SH2 domain. And so it has a binding pocket for the amino acid side chain and a binding pocket for the phosphotyrosine that allows it to plug into the receptor. So sometimes people get confused. It's the protein that's being recruited that has the SH2. This does not have the SH2. This has phosphotyrosine and the correct amino acid sequence. Different proteins have different ways of binding to phosphorylated tyrosines. SH2 is one way to do it. Okay? And so particularly in the RTK pathway, that's going to happen with several different proteins. They're going to stick to it. We'll talk about this one. PI3 kinase is an old friend of ours. It phosphorylates phosphatidylinositols on the membrane. Kind of convenient, right, that it's located, it's going to be attached right next to the plasma membrane. Because all it has to do is reach up here and find the nearest inositol that's got no, that has no phosphate group in free position, and that's what it's going to do. Okay, we'll get back to that one. This thing recruits an off switch for RAS that we'll get to, and we're going to talk about our old friend RAS. This is phospholipase C gamma. Remember, the one we were talking about was phospholipase C beta. This one actually is not up here in the membrane like the beta subunit. This is an isoform that binds to an RTK. So look at this. We've got this pathway that's going to be sending out signals. It's going to be sending out calcium signals this way. So this gets really complicated really fast. So let's deal with this one first. The RTK pathway, it's called the MAP kinase pathway, was first discovered in this little creature here. This is the Drosophila I. These are Ommatidia. They're photosensitive receptor cells. And so this is the way they're supposed to, if you take Dr. Hell's course, he'll talk endlessly about this, but there's a cell that's called R8 that's in the middle. And all these other photoreceptor cells, they have to touch R8 in order to survive apoptosis. Everybody else that's not touching dies, okay? And so in this mutant over here, this is missing, it's missing the R7, okay? And the reason it's missing the R7 is it does not have an RTK. And that RTK is supposed to be looking for a ligand that's displayed by R8. Okay. Since the receptor, the RTK, is not there, it can't stop apoptosis, and so it's missing a tooth there, right? And what does that do for this lovely fly? It means because it's missing R8, it cannot see ultraviolet light. And so that's how they screen for the mutation. The ones that could see ultraviolet light, they didn't have the mutation. The ones that were blind to ultraviolet light, like we are, couldn't see it. Okay? And so they were able to isolate this protein. And they call this, there it is, the RTK, it's called sevenless because it's missing the 7. And so they always keep cool names to their proteins. So that's sevenless. And This is the protein that was missing, an RTK. And is it supposed to dimerize? Yeah, it just doesn't show it, right? Okay, so there's some growth factor. In the case of the Drosophila, it's a protein that's displayed by RA. And that protein actually has a name. It's called BOS, bride of sevenless. I told you the names were cool. So bride of sevenless, if it's not there, and bride of sevenless is there, and you have no sevenless protein, the signaling pathway cannot be turned on, and so the cell kills itself. Okay, so how does this thing work? Well, you know how it works. It dimerizes, it cross-phosphorylates, it builds a bunch of phosphotyrosines, and then it's going to recruit this protein here. Okay, this protein is called GREB2. I spell out the name. It's growth factor receptor binding protein 2, since you're asking, but we call it GREB2. GREB2 is an adapter protein because it really doesn't do anything by itself. It's linking things together. It's a bridging protein. Okay, and so it's got an SH2 domain at one part of its molecule that allows it to bind to the activated receptor, and then it's got other domains called SH3 domains. What is an SH3 domain? You guessed it, Sarc homology 3. Okay, so if this is looking for a phosphotyrosine in a few amino acids right here, what is this looking for? It's looking for a part of a protein that has polyprolines. It binds the protein, and that makes it look really weird. Makes it be bent, has a bent tail, because it's got a proline there, and so it changes the direction of the peptide backbone, and that makes it look really juicy to an SH3 containing protein. So what this is going to do is GREB will recruit SOS. And SOS, since you're asking, is son of sevenless. This is BOS, the bride of sevenless. This is son of sevenless. Okay. And what is it? It is the GEF for RAS. So we finally get to what RAS does. We've been talking about RAS endlessly. You know that it's a lipid-anchored integral membrane protein. It's got two anchors. That's irrelevant at this point. When it's off, it's bound to GDP. It is one of those small GTP binding proteins. It needs a gap to turn itself off. But it also needs a GEF to be turned on. In this case, it's not a adrenergic receptor. It's a cytoplasmic protein called SOS. Okay? And in fact, this one doesn't show it. The SOS is even more intricately placed up here by the membrane. It's not floating around in the cytoplasm. It's actually up there close to the membrane for a reason I'll tell you. Okay, so what it does is it bumps into RAS, and RAS is converted to the active form. It loses GDP, picks up GTP, and it's going to completely, by protein-protein interaction, it's going to turn on the signaling cascade. Okay. Now, sometimes people get confused because we're going to talk about everything that comes next are all serine threonine kinases of some sort. This has no kinase activity. It's weird. It just bumps into things, just like the alpha subunit of a G protein. Okay. But remember, it's very small. It has no gap domain in it, so it has to rely on something like this. GTP, this is the gap. If it's a RAS gap, then it's going to turn that off. Okay, so what happens down here? Because we really haven't said what are the targets. This is the MAC kinase pathway. It's called the MAC-K pathway. And so here it is. Okay, so this is the horribly generic name for all these. Let's see, do I give you the, yeah, let's go back up here. All right, so here's RAS. It's going to bump into this protein. I'm going to call it by its nickname, which is called RAF. But the technical name is MAP kinase. MAP KKK. Okay? Because what it's going to do is when you activate this, it's going to phosphorylate MAP KK. You're losing a K every time, right? And so RAF phosphorylates MEK, and MEK phosphorylates the dangerous guy. This is MAP kinase. This is the one that's going to do all the things that cause this cell to go into cell division. Okay? It does a lot of things. Okay. What happened, a little bit impressive, is that this MEK is not only a serine threonine kinase, it's a tyrosine kinase. So this is probably I'm going to give you one more example in a couple of weeks, but these are rare. This phosphorylates MAP on two different residues. One is a threonine and one is a tyrosine. So it has two different kinds of activities. And so it's very careful to control MAP, because when you've committed to this, things are going to happen. Okay? This is the committing step in this pathway. And to get MAP turned on, you have to phosphorylate it in two different places, and then the binding lip changes so that it can receive its target. Its target is proteins, right? And it puts phosphates on serines and threonines. Okay? And it can go and target all kinds of different proteins that change protein synthesis activity, microtubule activity. In fact, the name MAPK did not come from anything to do with cell division. The first activity they noticed was to phosphorylate proteins that bind to microtubules. So microtubule attached or microtubule associated protein. That's what the A is. That's what a MAP is. Okay. Why do you need to reformulate all the microtubules in the cell if you're going to go through cell division? What's the normal, do you know the normal display of microtubules in a cell? We talked a little bit about it because all of the secretory pathways are all attached to the microtubules. They radiate out from the nucleus and go different places. Is that going to help you when you go, what has to happen when you go into cell division? You better have two poles, right? And they better separate, and they better have spindle fibers. You have to take all of your microtubules and reformulate them into a new array. That's what this guy does. And so that was the first activity, but now the M stands for mitogen activated protein kinase. That's what the M stands for now. But there are several different flavors of this. The one that's listed here is ERK, and extracellular regulated kinase is what that stands for. But MAPK is a really good general generic term. And so what it's going to do is it's going to, first of all, when you phosphorylate it, now it can dimerize. If it doesn't dimerize, it's not allowed to go into the nuclear form. Okay? It must dimerize. So that's another control level. So it can dimerize as long as it's got those two key phosphates, and now it can actually grab onto its target substrates. So it goes, one of those are, this makes lots of sense. It phosphorylates transcription factors that regulate gene transcription. Okay. So it's going to come in, and here's an old friend of ours, June, right? Remember what June was? We had FOS in June. They have a particular DNA binding thing, and it goes all the way back, so I would review that if I were you. But in order for this to sit on a response element, and there's a very specific response element that it sits on, it's called SRE, serum response element, because where are growth factors in all of us? They're all floating around in our bloodstream. And so the element of DNA that responds to that serum growth factor is a serum response element. And what sits on it is G, and then this thing here, ETS. Okay. But they can't sit there and do their job unless they're phosphorylated by MAPK. So that's the control. And that throws this thing into the cell cycle, which we're coming on very quickly in a couple of weeks. Okay, so now you know how transcription is activated by these growth factors. And MAPK does a lot of things that we'll talk more about some of the other things that it phosphorylates. But the two key ones are microtubules here and transcription factor. Okay. Questions? So get real familiar with that. You can just go receptor, Reb, sauce, RAS, RAF, MEC, MAP, and then you're done, basically. Okay. No questions? Very simple. Okay. All right. Why is it that this pathway is likely to be mutated in a cancer cell? Well, that's pretty straightforward. Any protein that, when it's mutated, will cause cell division is going to be an oncogene. And that's what these are. Okay. Now, a true oncogene is something that, when it's overexpressed or mutated, it causes cancer. Overexpressed or mutated. These are all normal proteins. Okay, oncogenes are all start out as normal proteins, like this, for example. Okay, MAPK is an oncogene. RTKs are oncogenes. So the receptor themselves, that is an oncogene. Okay? Because if you mutate it, for example, by say making this part look like it's bound to its ligand, so this thing is constitutively active, this cell is going to divide, and it's going to divide and divide. So that happens with the EGF receptor in breast cancer. All right? So anything that if you mutate it, it causes the cell to go to cell division. That's an oncogene. When it's still normal, what do you call it? A proto-oncogene. Proto-oncogene. Okay? So when you're looking through these pathways, ask yourself, well, is RAS an oncogene? Is it? Heck yes. 50% of the tumors that people have, RAS is mutated such that it doesn't even need sauce. It's turned on all the time. What about RAF? That's a big one, particularly melanoma. One of the new treatments for melanoma that works really well is a monoclonal antibody against that guy right there. What's the problem with that? I was just at a meeting in December, and the CEO of Genentech, they're the ones that developed that oncogene. The problem is that it just completely clears those people up. They look horrible with all these melanomas all over themselves. And then you give them that monoclonal, and it just completely cleans them up. 7 months later, right back. Because if you don't come down here and block some of these guys in addition, this thing's the tumor will become resistant. And that's actually how my PhD student developed a technique to block MEK, which is far enough down that the tumor really can't escape from it. And so that's now going to be in a clinical trial for cutaneous and T-cell lymphoma. So you can't just block these guys. You got to block these guys. And these are harder because they're intracellular. A monoclonal can come in here and bind to this, right, and block its activity. But you have to go further down the pathway or the tumor will become resistant. Okay, so that's a brand new strategy. Okay, so these are clearly all oncogenes. What's the other side of the equation? Well, cancer is really nasty because there's a bunch of other proteins that are called tumor suppressors. RAS-GAP would be a classic example of a tumor suppressor. Why? They're proteins that if you inactivate them or mutate them so that they don't work anymore, then you cause cancer. So you can imagine if we inactivated the protein that turns off RAS, and so the RAS-GAP no longer works. Let's say this. Let's think of a mutation. What if we mutated the SH2 domain of this RAS-GAP? Would that cause it to be qualified as a tumor suppressor? I mean, it is a tumor suppressor. Is that going to inactivate it? It has to, because it can't bind to the receptor. Why does it need to bind to the receptor to get close to RAS? Well, where is RAS? It's not cytoplasmic. So if this is floating around, the likelihood that it's going to bump into RAS is low. So RAS is up here. So if you recruit this right to the membrane, it's right next door. Okay? So that's why this is a good tumor suppressor, because if you mess with it and inactivate its activity, it causes cancer. It's just the opposite of what an oncogene does. Okay? So off switches to cell division are all tumor suppressors. Okay, and the other reason that RTKs are always seem to be messed with in tumors is that the scaffolding is so complicated that they recruit all kinds of signaling pathways. Okay? For example, you can have the RAS pathway going here, regulating MAPK. You can have phospholipase C gamma. The thing that distinguishes the gamma isoform is its SH2 domain. That allows it to stick to an activated RTK. What's dangerous about phospholipase C? What's downstream is really dangerous. What's the business end of all these pathways? Some kind of kinase. What's the kinase that turn on the calcium? Okay, I would look that up. All right, so the problem is you get all of these different kinases turned on that cause cell division, and it makes them very dangerous. Okay. All right, and so now let's talk about, this is also upregulated in cancer, unfortunately. In fact, that's why obesity makes women more likely to have breast cancer, just because if you upregulate the insulin receptor, then the tumor loves that fact. It loves to be upregulated. But so this signals a cell to divide, just like the rest of these RGKs. But what its main function is blood glucose regulation, right? Okay. So when insulins are activated, and you already know that it's a constitutive dimer, so it doesn't look like this. What insulin does is actually relieves the allosteric inhibition of, this is the alpha subunit, and this is the beta subunit. Okay? When there's no insulin, this is allosterically inhibiting the kinase. That's what's going on. So even though it's dimerized, it can't do anything because of the conformation of the alpha subunit. Insulin's job is to do one thing: change the conformation and relieve the allosteric inhibition. That's what insulin does. Okay? And so what happens is if you relieve that, then this becomes active. They autophosphorylate. That one phosphorylates this one phosphorylates that. And so you get this pathway instigated. It's different though. Okay. This part of it is the same, and what I wanted to show you is this is why SOS lives near the plasma membrane, because it binds to phosphorylated inositol head groups. Okay. And in order for that to be bind to SOS, it has to be phosphorylated at the 3 position. And do we know an enzyme that can do that? PI3 kinase is the one we've already talked about. It's recruited to this protein. So that becomes phosphorylated, that recruits SOS. You already know about GREB2. There's some other unnamed scaffolding protein that's going to recruit other pathways. And so you're going to turn on the MAPK pathway. So we know how that happens. What about the pathway to turn on the glucose transport? Okay, that's a different pathway. And it revolves around this enzyme. Okay, PI3 kinase, let's see if I show it. Yeah, PI3 kinase is recruited to the activated receptor because guess what? It's got an SH2 domain, right? SH2 domain. And that SH2 domain allows it to come close to the plasma membrane, and it goes around phosphorylating a lot of phosphatidylinositol cytoplasmic lipids, okay, at the 3 position. The reason that's important is that anything that has a so-called plextrin homology domain, pH domain, doesn't stand for potential hydrogen, it stands for plextrin homology domain, that allows these proteins to plug into a membrane lipid on the cytoplasmic side. Okay, so that's not an SH2, it's not an SH3, it's a pH domain. And so it recruits it to the cell surface. So this is a kinase, PDK1, goes up there. But this is the bad boy, all right, that is activated in the insulin receptor pathway. It goes up here, it binds to the cytoplasmic leaflet via its pH domain. And it's right next door to PDK. And these two guys are a coincidence game. You have to have this one activated, and you have to have mTOR activated. They're both serine threonine kinase. So the coincidence is they have to come and both phosphorylate AKT. AKT has another name. It's called protein kinase B, but we'll call it AKT, because most people in the literature do that. So this gets recruited to the membrane. It gets phosphorylated. That's the on switch. When it has both phosphates, it's active. It dissociates from the plasma membrane and goes around doing all this wonderful stuff with the GLUT4, right? If it phosphorylates so here's the pathway that we now know occurs. This was a bunch of hand waving when we talked about last time. You have the glucose transporter that needs to be inserted. right? So it's sitting there in what are called glucose storage vesicles, GSVs, waiting. What are they waiting for? They're waiting for the signaling pathway that we just talked about. PI3 kinase phosphorylates the plasma membrane, recruits AKT, PDK1, and TOR. AKT becomes activated, it phosphorylates this. This fuses with the membrane, and you know that story. One of these, we're going to talk mainly about this, but PI3 kinase phosphorylates the proteins that are blocking access of GLUT4 to the membrane. That's all you have to know about this slide. So PI3 kinase does some kind of miracle phosphorylation that causes vesicle trafficking to the plasma membrane. And when it does that, if those GLUT4 storage vesicles fuse with the plasma membrane, guess what? You've just delivered GLUT4, your blood glucose falls. That's the story. Okay. But AKT does dozens of things. One of those, there's two ways for cell number to increase in your body. One way is cell division. What's the other way? Doesn't involve cell division.
Keep cells from dying.
Exactly. Block apoptosis. Okay. And this is why insulin keeps cells alive. And this is why it's so dangerous to be overexpressed in tumors. Okay. So AKT, I guarantee, is an oncogene. It's a proto-oncogene, right? All right, so here's how that works. There's a protein that's called BAD, and it's a bad boy, all right? So what it's doing is it's inactivating a inactivator of apoptosis. So there are pro-apoptotic proteins that stimulate apoptosis, which causes the cell to die, and there's anti-apoptotic proteins. This little red thing is an anti-apoptotic protein. So if it's alive and moving around the cell, it blocks apoptosis. That's what the insulin signal says. I want you to block apoptosis. But it can't do that because of Mr. Bad. Okay? Mr. Bad ties up the anti-apoptotic, and if you block the blocker, apoptosis goes forward. So what the insulin receptor goes is, aha, I'm going to phosphorylate Bad, and then he'll let go of this protein. And that's what happens. So AKT phosphorylates Bad, and there's an inhibitory protein called 1433 that gloms onto it, and that releases this little red guy that blocks apoptosis. If you want to know the name, it's BCL2. It's a whole family. But we're not going to get into apoptosis. It's too complicated. Right now, we're just talking about how do you start it and how do you block it, right? So AKT phosphorylates Bad. It gets tied up with its little friend, 1433. So this thing can't possibly block this BCL2 protein. And this goes and stops apoptosis. The cell stays alive. So the number of cells are going to go up because they're not dying. The number of cells are going to go up because the MAPK pathway has been activated. Okay. That's a lot to digest. Questions about that? Yeah.
Are BAD and BAX interchangeable? Are they the same kind of?
They're in the same family. I just don't want to get too many monikers in there. So it's just leave it at that. But they're closely working together. Yes. Yeah. Okay. So this family is rather complicated. And so this is sort of the take-home message here. One of the reasons it's so difficult to defend against all these pathways is that they're all talking to each other, they're turning each other on, they're turning each other off. We've talked about a few ways of crosstalk between the calcium family and the MAPK family. So the upshot is you usually don't just get one, you get several. And the final output of this is difficult to predict. But this is a good review slide, because we've talked about every one of these pathways. Okay, questions? Okay, we'll start with the cytoskeleton. This is the end of quiz 3 material. And so I'm going to post the practice exam, get everybody started early, and then there's gonna be, okay, the other deal is, there's gonna be at least one more seminar that you can attend for credit. It's actually a person from Texas A&M that works on the signaling pathway, so that's a good way to earn 5 points. And I'm gonna put up all these assessments. They're not called assignments, they're called assessments. It's like a little quiz, and you gotta do stuff to be able to answer those questions. And those will go up by the end of the week. And the last thing you need to do is, that will be an assignment, it's gonna be, you have to rank your buddies, okay? And that has to be online. I'm gonna give you 5 points for doing that. Normally, I don't give anybody anything, but if you don't do it, we'll give you 5 points.